Hello, thank you so much, Gabby, and thank you to Hanson Wade for this opportunity to present our data. I'm Oscar Segurado, Chief Medical Officer of ASC Therapeutics, and I'm very pleased to introduce our company and also our work in gene editing and gene therapy for Hemophilia A. As an overview, a few aspects of our company: we have developed in-house gene and cell therapy platforms, starting with a cutting-edge gene editing platform called Target in collaboration with Stanford University, also in licensed CRISPR-Cas9. And we have developed a second-generation gene therapy and allogeneic cell therapy platforms. We have exclusive rights for CRISPR and gene therapies for Hemophilia A and B. The probably most relevant aspect of our company is that we are co-developing these technologies with top scientists and institutions worldwide, and that comes from over 12 years experience in gene editing and stem cell methods that were incubated at Applied StemCell. This company, the therapeutic aspect of this organization, really was working on all these aspects with a number of organizations and institutions such as Stanford, Karolinska, MIT, and interestingly, Emory University and Expression Therapeutics. And Gabriela Denning is the R&D VP for this organization. We license from them ET3. We're going to look into that in a moment. And we also got some licensing for gene editing through CRISPR Therapeutics. In our space, it's tremendously important to be able to be up to date in terms of the CMC manufacturing, quality systems, regulatory, and compliance. And we feel very comfortable because of our expertise and all these over a decade working as a CMO or CDMO and working with life sciences companies. We were able to establish a very close relationship with Vigene for AAV production and with City of Hope for cell therapy. We, based on all that and based also on all our experience, in my case, I have 25 years experience in clinical development. In the last 10 years, mostly focused on gene and cell therapy. So we were able to interact with FDA. We had pre-IND. We achieved 2 FDA orphan drug designations that enabled us fast-track approval and 7-year market exclusivity in the US. And in addition, we have a very experienced management team with all the options I just mentioned, with decades of experience in various stages of gene and cell therapy development. Here you see the basics of our platform for gene editing. And as I mentioned, we as a company, as a previous company, Applied StemCell, we were recognized as a leader in this space. Target is this proprietary technology that allows us, and that's the way I understand it, not being a real expert in the field, like placing a helipad in a very direct, specific site, and having a helicopter, which is whatever gene we want there, and which even a larger size DNA fragments, up to 20 kilobases. And to the right, you have the standard technology for CRISPR-Cas9, including knock-in, knockout, and being able to have a small and medium-sized DNA fragments. At the bottom, you see the work that we have been doing in our organization, placing the factor 8 in between exon 13 and 14, and very close to the safe harbor albumin locus, so that we are able to use that as a promoter, and in that way, with a second AAV, being able to insert that factor 8 and being able to produce factor 8. So this is the basic approach that we have to be able to produce factor 8 in vivo. And I think this is extraordinarily important because it's the way we can, for the first time, if everything works in the right way, with the pediatric population. And here you can see all the work that has been developed in our laboratories in terms of working with animal models with Hemophilia A. First, we started with the standard human gene, and from there we started moving into the ET3, some data that I'm going to show you in a moment. But our organization has very strong experience in terms of working with CRISPR-Cas9 and also different ways of integrating transgenes in mammalian cells. So now let's move into gene therapy. So we have and what I'm going to describe to you right now in terms of factor VIII gene therapy can be also applied to gene editing, because we use exactly the same construct except the promoter. That obviously the moment that you insert factor VIII into the somatic position, in that way you are using the albumin locus from the individual. In our case, in the case of gene therapy, we are using if you see the third bullet here, it would be a chimeric porcine human construct, which includes a number of sequences that were discovered and applied by the Emory University, and a number of individuals, scientists there, that were able to identify a B domain deleted transgene that was improving both the secretion and also the translation and folding of the protein. This is tremendously important, because the moment that we use this kind of factor VIII, it is going to be able to have a more efficient translation. That's what you see in the graph to the right, and a better folding and secretion of the protein. In that, when you have this kind of construct, you also need a special promoter, because you have increased through this chimeric construct the packaging capacity of the AAV, and it has also been part of what has been done together with codon optimized transgene. So we are very comfortable with this construct. We have also the experience also from Emory University and Expression Therapeutics that, as you heard, maybe some of you attended this morning, Gabi presentation with their work on lentiviral hematopoietic stem cell therapy, where they have transduced the 83 construct with a CD68 promoter. So we are using the same construct, but applied in this case to in vivo therapy through gene therapy. So this is different to all the data that has been produced by other companies working in the space. And this is the data that originally was generated by Emory and Expression, showing that the ET3 transgene has about 10 to 100 increase in terms of biosynthesis and secretion. And this is compared with HSQ, which is the regular human codon optimized B domain deleted gene that is used also by other companies that are also working in the space. Now, once we had this information, we started developing in our organization preclinical development aspects, IND enabling studies. All of these studies have been completed now. We are getting ready for an IND submission, and we could see also and confirm this increase in terms of production of the factor VIII, both in wild type mice, in humanized mice, and non-human primates. And finally, I just would like to show you how we are progressing in terms of our entire pipeline. That's what you can see here. In the middle, you see ASC 618. This is the program I just showed to you, where although we're working like all other companies in terms of working, of course, very closely with CROs and ensuring that we take into account all the information in the public domain in regards to long-term safety and assessment of capsid immunogenicity, potential carcinogenicity. All this data is serving us now to ensure that we have a very thorough monitoring of these patients. And in addition, we have a hypothesis, which is also has been discussed with Professor Stephen Pipe, he from the University of Michigan, and he is hypothesizing that the lower AAV dose of factor VIII would, and this improved biosynthesis and secretion, could potentially improve the durability of our gene therapy, in contrast to other companies working exactly in the same space. And then you see also there the ASC 518. This is this program that we have with the gene editing CRISPR-Cas9, and we're starting to develop that both in the US and for China. Thank you very much for your attention.
Thank you very much, Oscar. That was a wonderful presentation, and obviously we're familiar with ET3.